You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is September 20th, 2024. We will be discussing Chapters 6 and 7 of How the Immune System Works by Dr. Lauren Sompayrac. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine and Pediatrics and is the Chief in the section of Allergy Immunology.
So we can continue with our discussion for basic immunology. We are still on to our Lauren Sompayrac 7th edition discussion for board prep and just for clinical expertise. Here are my disclosures, nothing relevant to the talk today. The learning objectives for today are to improve our understanding on T cells at work and secondary lymphoid organs. So, again, September 20th, 2024, I'm Nikita Raje from Children's Mercy, Kansas City, University of Missouri, Kansas City. This is Chapter 6 and 7, discussion from Lauren Sompayrac textbook. So the Chapter 6 is T cells at work. So let's start that discussion. So we talked about how T cells get activated last time. So now once those are activated, we know that they form a clone, and then once they form that clone, those T cells differentiate. They differentiate into the effector cells. So if it's a helper T cell, it can make various types of subsets. And if it's a CD8 or cytotoxic T cell, it will make a CTL, effector T cell. So, kind of to understand the concept, think about, we had talked about how the T cells are the quarterbacks and they call the plays, right? So the helper T cells use cytokines or messenger molecules to convey that message and to convey their next steps. And they have several cytokines that they use so that they can make sure that the response is according to whichever infection is going on. So when they use particular type of cytokines, they set themselves as different subsets to use those cytokines, such as T helper cells subset 1, 2, 17, TH0, Tregs. So all these, let's see, all these are T helper cell subsets that have different functions. And then we talk about the CTLs as well. Now let's talk about these T cells and their coach, right? So the antigen presenting cells, like dendritic cells mainly for naive T cells, are their coach. And so how do the coach help those T cells make their decision? The way the dendritic cells convey the message and help out the T cells is when they are presenting the antigens to these T cells, they tell the T cells what type of invader they are dealing with and the site of invasion. How do they convey this message? The dendritic cells use their pattern recognition receptors to get the information from the environment of what type of invader they are dealing with. And then they use their cytokine receptors, because there is cytokine in the environment depending on the type of invader. So they use their cytokine receptors, and the type of cytokine receptors that get used tell their dendritic cells what kind of invader it is. So they have that information of the type of invasion. The site of invasion is also clear based on the cytokines that they are looking at. Whether these cells are in the intestine or skin, their cytokines that are released based on the invasion are different. Now the dendritic cells have the information, but they have to pass on that information to the T cells so that they can use it to make the decision of whether it's going to be the Th1 subset or Th2 subset, or which subset of the T helper cells are needed for that particular infection. And so the information transfer from the dendritic cells to the T cells occurs with the help of co-stimulatory molecules and the cytokines that are released. So they'll use the co-stimulatory molecules to help in activation of T cells, along with providing that information, as well as the cytokines that are released from the dendritic cells that act on the T helper cells also help in providing that information of the type of invasion and the site of invasion. So let's talk about each of these subsets, three main subsets, Th1, Th2, Th17. We'll also talk about Th0 cells, what's different about those cells. So let's talk about the Th1 cells. So the Th1 cells are produced or active in particular types of infections. So let's say the trigger is a virus in the cell or intracellular bacteria or a intracellular organism like mycobacteria that these cells have come into contact with. The dendritic cells will use their pattern recognition receptors and realize what kind of infection it is, and the cytokines that are released around them would most likely be IL-12. This kind of IL-12, in presence of these kind of cytokines that are released, these dendritic cells will be further activated to produce more IL-12. That IL-12 will act on the helper T cells to tell them that Th1 type of cells are needed. So they will provide that interaction with the Th cells to and further determine that these cells will become Th1 cells. These Th1 cells will release cytokines like TNF, interferon gamma, and IL-2. And these cytokines have particular functions that help in fighting the infection, such as viral infection or intracellular bacterial infection or mycobacterial infection. So what do these cytokines do? The TNF, we know, activates macrophages and NK cells. So guess what the macrophages and NK cells help in? They help in phagocytosis and cytotoxicity for intracellular infections. Interferon gamma is another cytokine that's released that keeps the macrophages activated. They also help in class switching the B cells to produce immunoglobulin G, particularly subclass 3. That helps in opsonization and complement activation. So the interferon gamma also helps in producing weapons that would be used to fight these particular infections. IL-2 helps in activating NK cells and prolong their killing function. They help in proliferation of NK cells, T helper cells, Th1 cells, as well as CTLs. What do CTLs help with? They also help in intracellular infection clearing. So all these cytokines have a particular function that is specific to kill the trigger that caused these cytokines to be released in the first place. What do I mean? So just kind of to recapitulate, at the site of infection, there were some sentinel cells like macrophages, mast cells, and dendritic cells. Macrophages release cytokines. Those cytokines and the patterns of the microbe were sensed by the dendritic cells. Once those were sensed, the dendritic cells go to the lymph nodes and on the way as well release IL-12. IL-12 acts on the helper cells, and these helper Th1 cells will release particular cytokines that are specifically going to help with intracellular infections. So those are TNF, interferon gamma, IL-2. Let's talk about Th2 cell. Here, let's think about an infection with a parasite. In that case, or if there is ingestion of pathogen, which means it's a mucosal infection, what happens? There is release of IL-4 in that environment. IL-4 will give that information. That information is taken by the dendritic cells, and the environment itself has IL-4, and all of that information is provided to the helper T cell, so the Th2 cells become activated, and Th2 cells release IL-4, IL-5, IL-13. What do these cytokines do? IL-4 is a growth factor for Th2 cells, so they help in their own production. They are also a growth factor for B cells, and they help the B cells class switch to IgE. Why is IgE needed? IgE is needed for clearing the infection by that parasite. IL-5 helps in class switching to IgA, and IL-13 helps in production of mucus, and that all of these help in clearing infections, mucosal infections, parasitic infections. So that's how the dendritic cells kind of orchestrate that play, where they take that information, provide it to the helper cells, and then the helper cells release the cytokines that were pretty much anticipated or by the dendritic cells. So they are kind of making sure that the cytokines that are released from that cell are specific to the infection that they saw in the environment where they were the dendritic cells. Let's talk about the third type, Th17 cells. Here there is, say, a fungal infection or an extracellular bacterial infection. In those cases, there is release of TGF-beta and IL-6. And when there are these two cytokines, a combination of these two cytokines, the T helper cells decide to become Th17 cells. So the dendritic cells take up that antigen, present it to the T cells, and the cytokines, in presence of the cytokines TGF-beta and IL-6, to cause the Th17 cells to release IL-17 and IL-21. What do these cytokines do? IL-21 causes the Th0 cells, which are uncommitted T helper cells, to become Th17 cells, and they class switch to IgG. So what does IgG do? It helps in clearing extracellular bacterial infections. Also, they help in IL-17 helps in neutrophilic recruitment, which is important for fungal infections as well as extracellular bacterial infections. IL-17 also helps in class switching to immunoglobulin G, and that also plays a role, as we saw in fungal or extracellular bacterial infections. Some uncommitted helper T cells can function in a little bit of a different way. So here, the dendritic cells will still act on these cells and activate them. They'll tell them where the infection is. Remember, two pieces of information that dendritic cell gives the helper T cells is where is the infection and what's the bug. In this case, it will inform the Th0 cells that where the infection is, but not specifically induce or give the information about what's the bug that's causing the infection. So these helper T cells will reach the site without being committed as Th1, Th2, or Th17 cells. When they reach that site of infection, say they find there is a lot of IL-12 in that environment, there are macrophages that are present, there is interferon gamma. In presence of that environment, cytokine environment, these Th0 cells will become Th1 cells. However, if there was a parasitic infection and there was a lot of IL-4, these Th0 cells would then become Th2 type of subset and help in that infection. So there is a role of these uncommitted or Th null cells in terms of getting to the site of infection and being flexible on their commitment to one subtype of the subset of the T helper cells. So let's talk about this profile of the helper T cells, where they get locked in to become either Th1 cells or Th2 cells or Th17 cells. So we know that when the Th subsets become committed to one type, either Th1 or Th2, there is a lot of positive feedback that they provide themselves. So when there are T helper cells type 1, they will use some cytokines to provide that positive feedback that they need more Th1 cells, for example, so that the uncommitted cells will become Th1 subset in that region. They also provide negative feedback to the other commitments. So they will try the TH0 cells do not become other subtypes. So they'll provide the growth factor to reduce the proliferation of other subsets. So for example, if there is interferon gamma that's released from TH1 cells, they will try to make sure that the cells do not commit to becoming Th2 cells because they need more Th1 type of subsets. If IL-10 is released, that will try and make sure that the subsets, the Th0 cells, do not commit to become Th1 cells. So there is a lot of negative feedback to prevent other subsets and positive feedback to make more of the same Th subset. Also, you have to realize that there is a lot of flexibility of simultaneous defense against different invaders in different parts. So say someone has a flu, but at the same time, someone has an allergic reaction to something. If all the Th subsets are just Th1 subset because they are trying to fight that flu virus, then what happens when there is some allergic reaction, or say there is a skin abscess and extracellular bacteria somewhere? You cannot have Th2 or Th17 subset of cells if there was no flexibility. However, these Th cells, there is a local action. So wherever they are needed, and I don't mean local by one site because they can be in the circulation, but there is flexibility that if there are other sites where there are other subsets needed, it's possible to have Th1 subset somewhere else working, whereas Th2 or Th17 that's working somewhere else. So that's how the Th cell profile is locked in. Now we talked about different types of Th subsets. Now those are all CD4 subsets, right? Now we talk about CD8 T cells. Now remember again, we are talking about CD8 T cells that got activated. So the naive T cells came across a particular antigen, a cognate antigen that was able to activate those CD8 T cells, and when they get activated, they proliferate, make a clone, and then differentiate. The CD8 T cells differentiate to become a CTL, or cytotoxic T cell. So the CTLs, effector CTLs, what do they do? How do they work? So their function occurs in the immunological synapse. So just as a refresher, we talked about immunological synapse, where the cell comes in contact with the T cell comes in contact with the antigen presenting cell. There are adhesion molecules, integrins, selectins that keep those cells closer to each other. And then there are other, the TCR that's binding to the MHC molecule with the peptide. And then there are other co-stimulatory and co-receptors that interact with the antigen presenting cell. So the whole area of that interaction is called immunological synapse. So when the effector CTLs are in action, they release these enzymes called perforin and granzyme B. What does perforin do? Perforin, as the name suggests, perforates and makes a hole. It makes a hole in the cell wall so that the enzymes that are needed for their action can be released into that immunological synapse. So the perforin can then make a hole in the target cell as well and release granzymes, particularly granzyme B, into that target cell, and that helps in enzymatic chain reaction in that target cell that leads to the apoptosis of that cell. That's one of the type of action that CTLs can use. However, there is another method of how the CTLs can perform their cytotoxicity. They have something called Fas ligand on their surface. When there is a damaged cell, that a target cell, it can express Fas on its surface, and the Fas can bind to the Fas ligand on the CTL, and that leads to the chain reaction that leads to apoptosis of the target cell. The vesicles are then cleared by the macrophages. So that's how the effector CTLs work. So we talked about the helper T cell and the cytotoxic T cell function. Now we can move ahead and talk about switching gears and talking about in chapter 7 about lymphoid organs. So any questions at this point? Right. We can move ahead here. We'll be talking about chapter 7, secondary lymphoid organs and lymphocyte trafficking. Again, today is September 20th, 2024, and we talked about T cell activation, antigen presentation, as well as the T cells at work. We also talked about B cells that help in providing the antibody function. Now we talk about why are these secondary lymphoid organs needed and how the lymphocytes move from one place to another. So just kind of a reminder, there are primary and secondary lymphoid organs. Primary lymphoid organs are bone marrow and thymus, where the lymphocytes are generated, and cells that are destined to become T cells mature in the thymus. Whereas the secondary lymphoid organs have a specific structure to it. The main structure that is present in secondary lymphoid organs is called lymphoid follicle, and we'll talk about those. But in general, to understand the need for these lymphoid organs and how the cells move, you have to think about the overall defense phenomena that occurs in our body. And there are 3 phases of it. Recognition of the danger. So we talked about how there are pattern recognition receptors and how the antigen presenting cells take up that information, present it to the T cells, or the B cells come across the antigen, and once that recognition occurs, they help in production of appropriate weapons. So we talked about the T cells activation, B cell activation, production of antibodies, production of effector T cells. And once that occurs, there needs to be the transport of those weapons to the site of attack. And that transport is important because otherwise there's no point of having all of that machinery there. So let's talk about the secondary lymphoid organ. So again, the main structure, important structure of the secondary lymphoid organs is a lymphoid follicle. And there is a structure called high endothelial venule in most of these secondary lymphoid organs except one organ. So we'll come to that information in these following slides. So let's talk about the lymphoid follicle. When you hear about lymphoid follicle, we are talking about B cells. There is a primary lymphoid follicle and a secondary lymphoid follicle. These follicles have follicular dendritic cells. These have nothing to do with the dendritic cells that we talked about with T cells. These follicular dendritic cells are completely different from those dendritic cells, and these are present in the follicle, which gives them the name follicular dendritic cells, and these take part in activation of B cells. So these follicular dendritic cells, or FDCs, are produced starting second trimester of pregnancy, and they are present in those secondary lymphoid organs. These are star-shaped, and that's how they get the name of follicular dendritic cells, and their job is to display the antigen to the B cells. So kind of similar to what dendritic cells do for T cells, where they are presenting those antigens to the T cells. Here, the presentation is a little different. Here they are just stationary. The follicular dendritic cells are present in the follicle, and they display the antigen on the surface where the B cells can interact with them. These follicular dendritic cells, so say this is a follicle, right, primary lymphoid follicle, and there are several B cells scattered around, and there are these follicular dendritic cells. What do? They have on their surface complement receptors. So say there is a antigen that is present in the circulation that comes across complement. Complement's activated, and complement helps in opsonization, so it tags that antigen. That tagged antigen with the complement fragment goes through the circulation and eventually ends up in the secondary lymphoid organ. there it's going to come to the FDC, and the FDC has a way of plucking that antigen and the because it has complement receptors, that complement opsonized antigen is going to attach to it. The other way the FDCs come across the antigens that they present is because they have Fc receptors. So antibodies that are later in the infection, when the antibodies are present, so the innate immune response has happened and the B cells are activated and the B cells can produce antibodies. Those antibodies end up in circulation, they come across the antigen and they opsonize that antigen. So the antibodies tag that antigen and eventually make their way to the lymphoid organ, so secondary lymphoid organ. There they enter the lymphoid follicle, and because the Fc receptors are present on these FDCs, they will bind that antibody opsonized antigen. Once they have that antigen either bound to the complement receptor or the Fc receptor, that antigen is present on the surface of the FDCs that can bind to the BCR. And because the multiple BCRs on the B cell can bind to those antigens, they can cross-link multiple BCRs and help in activation of B cells. When there is an antigen that can bind to a particular B cell and those B cells get activated, the primary follicles stop being primary follicles and develop a germinal center, a center basically it's in the center of the follicle, and it's called a germinal center. And when there is a germinal center present in that follicle, it is called a secondary follicle. So let's look at secondary follicle. Because the B cells got activated and they start proliferating. Those proliferating B cells proliferate at a very high rate. They double every 6 hours and they are pretty fragile, so they are very prone to apoptosis. But they multiply so fast that there is this whole center formed where these proliferating B cells are present, and that forms the germinal center as you see. Because these are proliferating B cells, because they have been activated by the antigen, they get rescued from apoptosis because they get help from T helper cells. When they get help from T helper cells, these B cells are going to be activated for a longer time. They are going to start to make plasma cells that leave the germinal center and produce antibodies. Now we know that during that time, B cells, this is the time where B cells decide if they want to be plasma cells, or over time they want to make more antibodies that are more of very fine-tuned to the antigen in question here. So they start undergoing hypermutation, somatic hypermutation in their FAB region to produce higher affinity antibodies. They also can class switch from producing IgM to producing IgG, A or E, depending on the type of infection. So as they go through these processes, they make a part of the germinal center that just have proliferating B cells, and they also have a region called light zone. That's the dark zone where there are proliferating B cells, and there is light zone in the secondary follicle where there are follicular dendritic cells expressing those antigens. So these proliferating B cells will go out to the light zone and get tested against that antigen, and the B cells that test good, so they pass that test, that they have higher affinity antibodies, will survive and the others will die. So over time there is improving affinity of these B cells for the antigen. And so once you have a good affinity, high affinity antibodies that are produced from B cells, again, they are differentiating into plasma cells, and the plasma cells will leave germinal center, and then they do their job of producing antibodies. All right, so that's a secondary follicle. So remember, primary follicle has follicular dendritic cells and B cells. When there is activation of B cells and the B cells start proliferating, they create a germinal center in that follicle, and then they are called secondary follicles. So now we talk about the second structure in the lymphoid follicles called high endothelial venules. All secondary lymphoid organs have high endothelial venules except for spleen. Where are these high endothelial venules present? They are present in the post-capillary venules. In the post-capillary venules, as you see, and as the name suggests, the endothelial cells are taller, they are high, that's the reason they are called high endothelial venules. And these high endothelial venules allow the lymphocytes to wriggle out of the blood circulation into the lymphatic organ, say a lymph node or any other secondary lymphoid organ. And these are the spaces between these HEVs are are places where tens of thousands of lymphocytes can wriggle out of the circulation each second into the lymphoid organ. So let's talk about each of these secondary lymphoid organ. The prototype of that being a lymph node. Let's look at how the lymph node structure is built. So for our purpose of discussion, the goal of the lymph node is for the lymphocyte to come in contact with the antigen and the antigen presenting cell, right? So for B cells, it's the antigen. For the T cells, it's the antigen presenting cells combined with the antigen. So our goal to understand is, where does the lymphocyte come from and where does it go out? Where does the antigen come through and how do they move in from one place to another in that lymph node? So look at this structure. It has the arteriole that's bringing in the blood. Then there are capillaries. There you will have the high endothelial venules, and then it turns into a venule and a vein. So the blood is going out. Also, there are incoming lymphatics that are coming into the lymph node. And these lymphatics, and the way the structure is, they pour into bigger spaces in the lymph node called sinuses. So initially, when the lymph is draining into the lymph node, it drains into something called marginal sinus. And then eventually it goes through the lymph node structure to go get into the medullary sinus, and then outgoing lymphatics use the coming out of the lymph node will carry that lymph fluid out of the lymph node. So as the lymphatic fluid comes into the lymph node, the cells in those or antigens know where to go, and some of them are pushed into particular areas. Similarly, the lymphocytes that are coming into the lymph node know where they are supposed to go in this lymph node. So there are two parts of, or two structures in the lymph node. It has to have a B cell area where the B cells know where to go, and the T cell area. So the B cell area is the follicle, as we just looked at, and those follicles in the lymph node are present in the structure called cortex. And next to it is called the paracortex, and that's where the T cells end up. So the paracortex is the T cell region of lymph node, and the cortex with the follicles is the B cell region of the lymph nodes. So now let's think about lymphocytes. Where are they coming from and where are they going? Lymphocytes, as we just talked about, enter the lymph node through the HEVs. They wriggle out from those venules into the lymph nodes. They also come in through the lymphatics. So the lymphocytes can come from two places, but they exit only through the outgoing lymph. Where is the antigen coming from? Antigen is coming through the lymph fluid using with the antigen presenting cells like dendritic cells, or because they are tagged by complement or complement fragment or antibodies. So the antigen is coming from the lymphatics. Lymphocytes are coming from either the lymph or the HEVs, and then the lymph T cells end up in the T cell region, and the B cells end up in the follicles. The flow of lymphatic fluid is from the cortex to the paracortex. And when the lymph enters these sinuses, there are these macrophages in the marginal sinus that will actually engulf and phagocytose some of the bigger antigens. So that's how the lymph node is structured. Now, how do we know how do these cells know where to go? How do the B cells end up in B cell region, T cells end up in T cell region? So it's a very orchestrated, again, very organized flow of the cells. And that is because of chemokines. So I think of chemokines and chemokine receptors as, just to kind of give you an idea, it's almost like GPS. When you drive a car, you enter, say you use your map, some kind of app where you enter the address. And when you enter the address and put it in, you are going to be told where to go. If it's a self-driving car, it's going to take you there, right? So these cells are actually self-driving cars. And once the address is entered onto them, those cells end up in that particular location. So let's look at how that works for the lymph nodes. So lymph nodes have the lymphoid follicles that have follicular dendritic cells. Follicular dendritic cells release CXCL13, a chemokine that attracts naive B cells. The naive B cells, that's the reason when they enter the lymphocytes enter the lymph node, the naive B cells will be attracted towards the follicle because they are moving towards the CXCL13. If they get activated, if a B cell gets activated, it will actually start downregulating CXCL13 receptors and upregulate CCR7 on their surface. These activated B cells will, using the CCR7, will end up closer to the T cell region. So any cell that expresses CCR7 on their surface will go towards the T cell region. So the naive T cells express a lot of CCR7 and they end up in the T cell region. When the helper T cell gets activated, it actually starts down-regulating CCR7. So they move away from the T cell region and go closer to the B cell region. That's where at the border of B and T cells is where the activated B cell and activated T cell will meet so that they can interact with each other, and the T helper cells can help B cells in their job, whether it's for somatic hypermutation or class switching. So CXCR5 is the receptor on the B cells that attracts them towards the CXCL13. When the cell expresses CXCR5, initially naive B cells, they go towards the B cell region. When the T cells express CXCR5, once they are activated, they end up closer to the B cell region again. If a cell expresses CCR7, like the naive T cells, they end up initially in the T cell region. If they get activated, they down-regulate the CCR7 and then end up in the closer to the B cell region. So cells crawl towards the chemokines. Helper T cells once they are at that border of T and B cell region, the B cells and T cells interact with each other. Follicular dendritic cells express that antigen and present that antigen to the T cells. T cells use that information and express the co-stimulating molecule CD40 ligand that interacts with CD40 on B cells, and these cells will activate each other and help TH cells will help the B cells in their job. When that interaction occurs, there is a role for co-stimulating molecules other than CD40 ligand, such as B7 that interacts with CD28 on the helper T cells, and B cells also express ICOS ligand. Inducible co-stimulator is ICOS ligand that interacts with the ICOS on TH cells. These follicular helper cells, TH cells, will rescue the B cells from apoptosis, help in class switching and somatic hypermutation. If the plasma cells are formed, they will initially produce IgM, but once there is help from follicular helper cells, they will help produce more class switched. They'll have the class switching and produce other antibodies. The antigen is the same for both these cells, but the epitope that they use to get activated is different for B cell and helper T cell. All right, T cell recirculation, the naive T cells in the helper cell, in the blood, naive T helper cells go to the lymph node as we talked about. And if they do not come across an antigen and do not get activated, they re-enter the blood. If they do come across an antigen that they get activated by, the activated T helper cells stays in the lymph node for longer time, proliferates, and there is increased activation due to presence of dendritic cells. Once they do that, they exit the lymph node, they re-enter the activated T cell will then get activated and then proliferate, and then that will cause the activated T cell to go to the site of infection. However, if it's similar to that, the naive CTL will be present in the lymph node in the T cell region, and if it gets activated, it will proliferate and then recirculate and enter the tissue to kill the microbe at the site of infection. The other type of secondary lymphoid organs are Peyer's patches. Peyer's patches also have HEVs where the lymphocytes enter, but there are no incoming lymphatics. There is no antigen coming in from lymphatics here. There are only outgoing lymphatics. The antigen is coming to these Peyer's patches through M cells. So what are these M cells? M cells do not have villi, but they are present just at the tip of that lumen of the intestine, and they come across all the antigens that's coming into the intestine. There's no mucosal coating there. They bind to the selective antigens and transport those antigens from lumen to the tissue, and then from there to the draining lymph nodes. There might be complement fragment or antibody opsonized antigens that will be used, and once they get those antigens, they are again used by FDCs in the lymphoid follicles. So again, here is the place where there are HEVs. There is a lymphoid follicle, which is the B cell region, and the adjoining T cell region where the B cells and T cells end up. And you see the M cell here on the top of the Peyer's patches. Spleen is another one. It's a blood filter. 5% of the cardiac output is moving through the spleen all the time. There are no HEVs. So the lymphocytes are just entering freely. It's a blood filter. So the blood is just flowing freely. And so the lymphocytes are entering spleen all the time. They do not need HEVs. There are, again, no incoming lymphatics because it's the blood that brings in the antigen. So the artery is moving through, the splenic artery goes through the spleen. It goes through the marginal sinus. Here is the marginal sinus. And then it forms the vein and gets out of the spleen. There are macrophages in the marginal sinus that will take up the bigger antigens. And then the lymphocytes are entering the B cell region and the T cell region. So the B cell region have the lymphoid follicles, and along the arterioles are the T cell regions, and they are called periarteriolar lymphocyte sheath or PALS. That's the T cell area of the spleen. All right, so that's where the spleen works. Again, just to kind of sum it up, function of secondary lymphoid organ is just that they are in strategic position where they are conducive to the mobilization of the appropriate weapons and interaction between different cells. And different adhesion molecules help in deciding where the cells go, such as the interaction between naive lymphocytes, which is non-restricted, going from one lymph node to another. They use L-selectin and glycam-1 interaction of chemokines, whereas for particular place like the GI tract, there might be particular adhesion molecules like alpha-4 beta-7 interacting with MADCAM-1. So that's an important fact to remember. Compared to naive T cells, activated or effector lymphocytes have restricted movement, which means that cells that got activated in the gut will home back or go back to the gut or the skin where they got activated. Those lymphocytes will go back. They have restricted movement going back to the skin. And so there are particular adhesion molecules that will help to do that. For inflamed mucosal blood vessels, the different adhesion molecules that are important are alpha E, beta 7, and that interacts with different adresins. All right, that's the end of chapter 6 and 7. We'll talk